Hello friends, welcome back to Bio One Education. In episode 5, we followed water's journey through the plant. Today we ask what else does a plant need beside water? Welcome to Mineral Nutrition. We will cover essential elements, deficiency symptoms, the nitrogen cycle, biological nitrogen fixation, and finish with the exam tricks and animations. Let us begin. This chapter scores 2 to 3 questions most year and many are pure recall. They name an element and ask its role or describe a symptom and ask which element is deficient. The nitrogen cycle is probably the most tested diagram in all of botany. My suggestion for each element make a flashcard with its role on one side and its deficient symptoms on the other. For the nitrogen cycle, learn the bacteria at each step. That is enough to answer almost every question. How do we even know which minerals a plant needs? The answer is hydroponics, growing plants in a nutrition solution instead of soil. The idea is simple but powerful. You prepare a solution containing all known minerals, then remove one element at a time and watch what happens to the plant. If the plant gets sick, that element is essential. Julius von Schack pioneered this technique in 1860. Arnon and Stout later set three criteria for an element to be called essential. It must be absolutely required for the plant to complete its life cycle. It cannot be replaced by any other element, and it must be directly involved in metabolism, not just needed indirectly. Plants need 17 essential elements. They split into two groups. Macronutrients are needed in large amount. Nine of them, carbon, hydrogen, and oxygen, come from air and water. Nitrogen, phosphorus, and potassium are the big three that farmers add as a fertilizer, and calcium, magnesium, and sulfur play structural and enzymatic roles. Micronutrients are needed only in traces, but they are equally essential. Eight of them: iron, manganese, zinc, copper, boron, molybdenum, and chlorine, also nickel. Most work as enzyme activators or cofactors. A common exam trick: micro does not mean less important, it means less quantity. Remove any one of them and the plant dies. Let us meet the key macronutrients and their roles because NEET loves asking exactly this. Nitrogen is part of protein, nucleic acid, and chlorophyll. When it is deficient, older leaves turn yellow first, that is chlorosis, and it starts with older leaves because nitrogen mobile and the plant moves it to younger leaves. Phosphorus is in ATP, nucleic acids, and phospholipids. Deficiency gives leaves a purple or dark green color and delays maturity. Potassium regulates stomata, activates enzyme, and maintains osmotic balance. Deficiency causes scorched or burned leaf margin. Calcium is in the cell wall's middle lamella. Deficiency stunts roots and deforms a new leaves. Magnesium sits at the center of every chlorophyll molecule. Its deficiency causes interveinal chlorosis. Yellowing between the veins while veins stay green. That is a classic exam image. Sulfur is in certain amino acids like cysteine and methionine. Its deficiency looks similar to nitrogen deficiency but appears in young leaves first because sulfur is less mobile. Now the 8 micronutrients. Tiny amounts but remove any one and the plant is in trouble. Iron is needed for electron transport and chlorophyll synthesis. Its deficiency causes intervenal chlorosis in young leaves, not old, because iron is immobile. Manganese helps split water. Manganese helps split water in photosynthesis. Zinc is needed for axine synthesis. Its deficiency causes little leaf disease. Copper is part of cytochrome oxidase. Boron is critical for pollen germination and sugar transport. Its deficiency causes hollow stems. Molybdenum is a favorite exam element. It is part of nitrogenase, the enzyme that fixes nitrogen and also nitrate reductase. Its deficiency in cauliflower causes Whiptail disease. Chlorine helps split water in photosynthesis, and nickel, the most recently added essential element, is part of urease enzyme. When a mineral is missing, the plant speaks through visible symptoms. Four key ones. Chlorosis, yellowing of leaves because chlorophyll cannot be made. Many elements cause it: nitrogen, magnesium, iron, manganese, and more. Necrosis, actual death and browning of tissue. Calcium, copper, and potassium deficiency can cause this. Stunted growth. The plant stays small, low nitrogen or micronutrients, slow cell division. The trick for the exam is knowing where on the plant symptoms appear first, and that depends on whether the element is mobile or immobile. Let us see the next. This slide is an exam gold mine. If a question describes where on the plant a deficiency symptoms appear, you can immediately narrow down the element. Mobile elements like nitrogen, phosphorus, potassium, and magnesium can be moved from older leaves to younger ones. So, when they are deficient, the plant sacrifices old leaves first. Symptoms appear in older leaves. Immobile elements like calcium, iron, boron, and copper cannot be moved once they are deposited. So, deficiency symptoms appear in young leaves and new growth because the element is stuck in the old tissue. One rule: old leaves affected means mobile element, young leaves affected means immobile element. This single shot answers many NEET questions instantly. Can a plant have too much of minerals? Absolutely. Any mineral ion in excess becomes toxic. Toxicity inhibits enzyme activity and creates ion imbalance. The tricky part is that excess of one ion can block absorption of another, causing symptoms that look like a deficiency of that second element. The textbook example is manganese toxicity. Too much manganese causes brown spot on leaves and blocks the absorption of iron, magnesium, and calcium. So the plant shows their deficiency symptoms too. This concept is called ion antagonism, and NEET has asked it. The nitrogen cycle overview. Nitrogen is everywhere. 78% of atmosphere is nitrogen, but plants cannot use it directly. It must first be fixed, converted into ammonia or nitrate before roots can absorb it. The nitrogen cycle has five key steps. Fixation. Nitrogen is converted to ammonia by bacteria like Rhizobium and Azotobacter, or by lightning. Nitrification. Ammonia is converted to nitrite by Nitrosomonas, then to nitrate by Nitrobacter. Assimilation. Plants absorb nitrate and build amino acid and proteins. Ammonification. When organisms die, decomposers break organic nitrogen back to ammonia. And denitrification. Bacteria like Pseudomonas convert nitrate nitrate back to atmospheric nitrogen, closing the loop. For the exam, the bacteria names at each steps are the most tested facts. Nitrosomonas and Nitrobacter for nitrification, Pseudomonas for denitrification, and Rhizobium for fixation. Here is the nitrogen cycle as a looping animation. Watch each node light up as it becomes the active steps. Watch each node light up as it becomes the active step. Fixation, nitrification, uptake, then ammonification, return to nitrogen. Ammonification returns nitrogen to ammonia and the cycle repeats. For revision, narrate the bacteria at each step as the islet moves. Rhizobium fixes, Nitrosomonas and Nitrobacter nitrify, plants absorb, decomposers ammonify, and Pseudomonas denitrifies. One loop covers the whole cycle. Biological nitrogen fixation. Now the most tested topic in this chapter, biological nitrogen fixation. The enzyme nitrogenase converts nitrogen to ammonia using a massive amount of energy, 16 ATP per molecule of nitrogen fixed. And here is the critical exam fact. Nitrogenase is irreversible destroyed by oxygen. So it must work under strictly anaerobic condition. The most important organism is Rhizobium, which lives in root nodal of legumes in a symbiotic relationship. To protect the nitrogenase from oxygen, the nodal produces special protein called leg-haemoglobin that scavenges oxygen and keep the environment anaerobic. Other nitrogen fixers include Azotobacter, a free-living soil bacterium, cyanobacteria like Anabaena and Nostoc, which can be free-living or symbiotic, and Frankia and actinomycete that forms root nodal in non-legumes like Casuarina. Here is the nitrogenase reaction animated. Watch the nitrogen molecule approach the enzyme, bind to the molybdenum iron active site. 16 ATP get consumed to break the incredibly strong triple bond and two ammonia molecules pop out. Notice the labels. Anaerobic condition on one side, leg hemoglobin protecting the enzyme on the other. Those two details are almost guaranteed exam points. Time to gather every trick into one card. Screenshot this. Magnesium is the center atom of chlorophyll. Molybdenum is in nitrogenase and nitrate reductase. Symptoms in old use means a mobile element, nitrogen, phosphorus, potassium, magnesium. Symptoms in young leaves means immobile element, calcium, iron, boron. Nitrogenase uses 16 ATP per nitrogen molecule and leg hemoglobin shields nitrogenase from oxygen in root nodule. Let us step outside the textbook. The Green Revolution was powered by synthetic NPK fertilizer, nitrogen, phosphorus, potassium fertilizer. But overuse is now causing soil degradation and eutrophication of water bodies. A cautionary tale about mineral nutrition gone wrong. Biofertilizers like Rhizobium, Azotobacter, and Mycorrhiza offer a greener alternative. They are cheaper, eco-friendly, and enrich the soil. Scientists are now trying to engineer nitrogenous genes into cereal crops like rice and wheat. If they succeed, these crops could fix their own nitrogen, ending our dependence on synthetic fertilizers. And here is the fun connection. NASA uses hydroponics, the same technique from 1860, to study how astronauts might grow food on Mars mission. Your textbook technique is literally space technology. Surprise round. NCERT hidden lines that became questions. In elements, magnesium is the center of chlorophyll. Sachs pioneered hydroponics. Arnon and Stout set the essentiality criteria, and molybdenum deficiency caused whiptail in cauliflower. In the nitrogen cycle, nitrogenase needs anaerobic conditions. Nitrosomonas and Nitrobacter do nitrification. Pseudomonas does denitrification, and Leg-haemoglobin is found in the root nodule. More gems. Manganese toxicity induces deficiency of iron, magnesium, and calcium. Zinc deficiency causes little leaf. Boron is essential for pollen germination. Nickel is the most recently added essential element, and the Azola Anabaena association is used as a biofertilizer in rice paddies. NEET asks this chapter in four costumes: assertion and reason. For example, assertion. Nitrogenase required anaerobic conditions. Reason oxygen irreversibly inactivate the enzyme. But true. Reason explains the assertion. Match the column. Bacteria with their steps are elements with their roles. How many are correct? Mixing up element roles are deficiency symptoms. Diagram-based labeling steps in the nitrogen cycle are identifying which deficiency matches the symptoms photograph. Rapid fire. Pause. Answer in your head, then play again. Center atom of chlorophyll, magnesium. Bacteria for nitrification, Nitrosomonas and Nitrobacter. Nitrogenase needs how many ATP? 16. Whiptail disease is caused by deficiency of molybdenum. Mobile element deficiency shows symptoms in which leaves? Older leaves. And leg hemoglobin protects what? Nitrogenase from oxygen. Four or more right, you are exam ready. Let's wrap up. Final challenge. Which protein protects nitrogenase from oxygen in root nodule? Drop your answer in comment. Thanks for learning with me. See you next session.